Okay, so welcome back, everybody. And as you might have seen, last week I was unable to take my scheduled session. Instead, I took that session on Friday. Many of you wouldn't have joined that, I perceive, but I have recorded that session as well, and I'll soon upload the recording for that. Maybe tonight only I'll upload for that particular session. What we began discussing in that, apart from week seven's previous year questions, yeah. So apart from week seven's previous year questions, what we began discussing in that previous session was, about learning and memory and what is exactly, what are the kinds of memories, what is a short-term memory, what is long-term memory, what is the perceived notion about it, which might not be correct actually, and how they are stored is what we are going to study today in the second part of this session. So, but what are the modalities related to learning and memory? What are the kinds that all like that is what I had taken in the previous session on Friday. So I'll upload the recorded lecture for that as well. And today, in the second half of this particular discussion session, after the previous year questions, what we'll be discussing is how exactly those memories are, getting how they are getting formed is what we have studied earlier, how they are getting stored, or how that consolidation is happening from short term to long term. What are the processes behind it? What is the molecular level mechanism behind it? Is what we are going to look at, right? Um, okay, so let us currently begin with the previous year questions for week 8. The first one, so week 8, you might have already gone through. It has more to do with the various kinds of drugs, various kinds of, theoretical neuroscience. So eventually, during our discussion, we'll come to theoretical neuroscience also. We'll come to these drugs also. But before that, we have a few topics that still need to be, discussed. One is learning and memory, which is an ongoing discussion. After this, first, I would like to discuss probably the methods or the techniques that are used to study or assess the brain. And post that, probably we'll come to different kinds of disorders, different kinds of medications for those disorders. And finally, because I am myself not a pro or not very learned also in the field of theoretical neuroscience, so eventually we'll come to that. Probably it'll give me also some time to go through that concept a bit more, right? So currently, we'll discuss about these medicines, some of the questions regarding the theoretical neuroscience, right? So that is what this week's previous year questions pertains with. So, who discovered chlorpromazine? Chlorpromazine, right? So, who was the scientist who discovered this particular drug? Was it Pierre Deniker or Jean Delay or Henry Lebroyd or Paul Charpentier, right? So, which one of them actually discovered this drug? It was Henry Lebroyd, right? So, Henry Lebroyd, you can see his image on the right? This is Henry Lebroyd. Chlorpromazine is actually a phenothiazine, right? So it is the under a class of compounds which are known as phenothiazine. It is used to treat psychotic disorders such as schizophrenia or manic depression in adults. Manic depression, also known as bipolar disorder, right? What is the mechanism of action of this particular drug? It primarily acts as a dopamine D2 receptor antagonist. What does an antagonist mean? That it will go and uh, instead of dopamine, like it will block the receptor somehow, right? So with additional blockade of serotonergic, histaminergic, muscarinic, and alpha adrenergic receptors, producing both therapeutic as well as side effects. So primarily it is an antagonist for this dopamine receptor, but also it does have off-target blockade for serotonin receptors, histamine receptors, and other such neurotransmitter receptors also, right? So that is why, although it is therapeutic, that is, it has that calming effect. It, slows these people's brains down so they don't feel as agitated, probably. But it does have side effects, right? Because all these neurotransmitters are actually required in our normal physiology as well, right? So if we are blocking their release, it will lead to some side effects at least, right? What effects does razorpine have on patients with schizophrenia, right? One, it causes hallucinations, or it has a calming effect, or it increases the anxiety of these patients, or it has no effect at all. So, Rizapine, if you remember this discussion, was actually a hypertensive drug, right? So, it was a natural a drug that was derived from a plant, and it was actually originally used for hypertensive patients, right? To treat hypertension. So this is Rizapine. But later it was seen that it also has a calming effect in patients who were undergoing psychotic episodes. So it was given to patients with schizophrenia and it had a calming effect on these patients, right? So what is exactly Rizapine? It is a medication which is primarily used to treat high blood pressure or hypertension, as we just spoke about. It works by slowing down the nervous system, which helps relax the blood vessels and improves the blood flow. Rizapine is also used to treat agitated psychotic conditions such as schizophrenia, as we said in the previous statement. Also, reserpine works by irreversibly inhibiting the vesicular monoamine transporter VMAT, right? Depleting norepinephrine, dopamine, and serotonin from the nerve terminal, which reduces sympathetic nervous system activity and lowers blood pressure. So, what it does is the why it is uh, a little alarming to be used as a drug is because it is irreversibly inhibiting the formation of certain of these vesicles, which will go and carry the neurotransmitters. So it is irreversibly going to deplete at least some pools of these neurotransmitters from the nerve terminals, right? So, this would not just lead to that calming effect that we see in psychotic patients, but in normal patients without who do not have psychosis probably and who do not have an hyperactive or hyper alert brain state, right? For them, it could actually lead to some devastating effects also, right? What has replaced tricyclic drugs in treating depression? So, is it MAO inhibitors or SSRIs or antibiotics or pain relievers? Right? So, the most commonly used or that you might have heard of a class of drugs is actually SSRIs right now for treating depression, right? So, they do not have a lot of side effects also. That is one key point, which is why they are used also very much, right? So what this SSRI is selective serotonin reuptake inhibitors, right? So it is selective for serotonin reuptake itself, right? So what generally happens is, oh, first let us read this, then I can explain. So these SSRIs are type of antidepressant prescribed most often. They can ease symptoms of moderate to severe depression also. So we know that serotonin is one of the many chemical messengers. So it is also a neurotransmitter in the brain, right? Neurotransmitters carry signal between nerve cells in the brain, which are known as neurons. After carrying the signal between the brain cells, serotonin, what generally happens is, for instance, there is a synaptic cleft that is present. Neurotransmitter is released from the presynaptic neuron. It goes into the synaptic cleft and it is uptaken by the postsynaptic neuron. There are receptors on the postsynaptic neuron which uptakes this neurotransmitter, and it further transfers the message that was to be carried, right? But now, not all or not 100% of whatever neurotransmitter was there in the synaptic cleft would be, taken up by the postsynaptic neuron, right? So what happens? Whatever amount is left, it needs to be, removed from there, or it needs to be destroyed, or it needs to be recycled. At least over there, it should not be present, because we don't want a constant stimulus that is present, which will keep exciting the postsynaptic neuron, right? We need to give them that intermediary rest also. So what happens is, after this neurotransmitter has done its activity, after a certain point of time, what happens is, there are different mechanisms to remove the neurotransmitter that is left over in the synaptic cleft. In case of acetylcholine, for instance, we had studied that there is acetylcholine esterase, which is an enzyme which degrades acetylcholine, which is left over in the synaptic cleft, right? In case of serotonin, what happens? There is no degrading enzyme that is present. Instead, there are certain receptors in the presynaptic neuron, right? What these receptors do is they open after some time of the release of serotonin, and once they open, they again reuptake whatever serotonin is actually left behind in the synaptic cleft, so that it can be reused once another, signal needs to be sent, right? It can be re-released in that way. So, what SSRI does is it inhibits the reuptake of this serotonin, right? So after carrying a signal between brain cells, serotonin is usually taken back into those cells, which is a process called as reuptake, right? But SSRIs block this process. The blocking reuptake makes more serotonin available to help pass messages between the brain cells. Serotonin reuptake inhibitors are called selective because they mainly affect only serotonin and not other neurotransmitters, and they may be used to treat conditions other than depression also, such as anxiety disorders. Okay, wherever serotonin has a role to play, it can be used as a treatment strategy, right? So generally in depression, what these patients were suffering from was, I'm talking about the most wide scenario. Most of the patients, what happens is there is a less amount of serotonin that is being released, right? So there is either it is not being released or it is not being uptaken by the neuron. So there is some kind of desensitization or chemical imbalance which is happening, and generally it is coming because of serotonin, right? So if the serotonin level is maintained over there, then it can help relieve the symptoms of this depression also, right? So that is the principle behind it. So what type of a drug is meprobamate, right? Is it a type of food or an antibiotic or a painkiller, or is it a minor tranquilizer, right? So actually it is a fourth one, that is it is a minor tranquilizer. This is the uh, structure of meprobamate. So this particular drug is a carbamate derivative which is used as an anxiolytic drug, means to treat anxiety it is used, right? It was the best selling minor tranquilizer for a time, but has largely been replaced by benzodiazepines. So benzodiazepines such as Valium, right? Due to their wider therapeutic index and lower risk of toxicity at therapeutically prescribed doses, and lower incidence of serious side effects as well, right? So, what neurotransmitter do these benzodiazepines affect, right? these widely used drugs such as Valium, which is the neurotransmitter that they are going to go and affect? Is it serotonin or dopamine or GABA or adrenaline, right? So actually, these particular neurotransmitters, they go and the hint is they affect an inhibitory neurotransmitter, right? So which one will it be? It will be GABA, right? So this is the structure of benzodiazepine. Um, Benzodiazepine enhances the effects of GABA and at the GABA A receptors. It is an inhibitory neurotransmitter, so inhibitory effects will be enhanced, right? Increasing chloride ion influx and reducing neuronal excitability. So, what is the mechanism behind it? Benzodiazepines are GABAergic agents that act on the central nervous system to produce sedative, anxiolytic, anticonvulsant, hypnotic, and muscle relaxant effects. They do not activate the GABA A receptor but function as the positive allosteric modulators. What does that mean? So direct interaction is not there with the active site, but somehow they will indirectly modulate their active site so that um, binding to a specific site located at the interface of alpha and gamma subunits of the receptor, this binding enhances the effects of endogenous GABA and the primary inhibitor neurotransmitter in the CNS, right? Sorry. So, what term did Selye replace alarm reaction with, right? So, was it anxiety or stress or adaptation or hormones, right? So, the answer is simple, it was stress. So, Hans Selye, he was a person who extensively worked on and described what stress is, right? Still, it is an ambiguous term till date because stress could be oxidative stress, cellular stress. Uh, Different kinds of stressors could induce different kinds of stress, right? This was a statement that he made in 1980. Stress like relativity is a scientific concept which has suffered from the mixed blessing of being too well known and too little understood, right? So every other person that you will meet would have encountered stress, some or the other kind of stress at this point, right? So kids aside, most of the adults would have encountered some or the other kind of stress by now, right? So everybody knows that there is something known as stress. Everybody knows the feeling of what it is, but we don't really understand exactly what it is. Like I said, depending on the stressor, the stress could be different, right? It could either be some stress in a particular kind of organelle or another kind of stressor producing another kind of stress in another organelle altogether, right? So we don't know. What did Claude Shannon optimize using, electromechanical relays? Was it Boolean algebra or action potentials or neuronal connections or psychological disorders, right? So again, this is part of that theoretical neuroscience unit that had been introduced to you, and the correct answer for this is Boolean algebra, right? So this is Claude Shannon, and Boolean algebra is what he optimized, right? In his thesis, Shannon proved that Boolean algebra could be used to simplify the arrangement of relays that were the building blocks of electromechanical automatic telephone exchanges of the day. He went on to prove that it should also be possible to use arrangements of relays to solve Boolean algebra problems. His thesis actually laid the foundation of for all the digital computing as well as digital circuitry that is currently there, right? So that was the work of Claude Shannon. The utilization of binary properties of electrical switches to perform logical functions such as that and, or, not, these gating, right, is the basic concept that underlies all electronic digital computer designs, right? So if even back in school, if you have ever, studied these logical terms, how, what they are, right? The if-else statements, the or gate, right? So that all came into existence because of his work, which was the foundation of it all. Shannon's thesis became the foundation of practical digital circuit design when it became widely known among the electrical engineer community during and after the World War II, right? So that was the era in which he actually worked. So, what is the main purpose of the ENIAC computer? Is it to play games or to compute ballistic trajectories or to store pictures or to write letters? So, back in the day, storage problems came much later once processing started, once the kind of computing power had already been established. Did we actually need to store whatever was computed, right? So that was not a problem. It was not developed to play games also. And it was definitely not to write letters. There was a typewriter for that. We didn't need a computer for it, right? It was actually proposed during the time of World War as a powerful device which could compute ballistic trajectories, right? So those missile, how accurate and from where, which coordinates would the launches be more accurate, all these things. So that was what it was proposed to do. So ENIAC, when we expand it, it is electronic numerical integrator and computer, right? So you can see that whatever it can do is in the name itself, right? In the expanded name. It was the first programmable general purpose electronic digital computer which was completed in 1945, right? At the time of the World War II. And it marked a significant milestone in computing history. ENIAC was developed during World War II primarily to calculate the artillery firing tables for United States Army. The project was led by John Macaulay and J. Presper Eckert at the University of Pennsylvania's Moore School of Electrical Engineering with funding from the US government. Then came its successor, EDVAC, right? So, what was the key feature of EDVAC? Was it that it has no memory, or it was made of wood, or it operates at high speed without any logic whatsoever, or it used stored programs for computations, right? So, which one was a key feature of EDVAC? Now, let us go through this. Does something having no memory shout out feature do you know? So this we can anyway rule out, right? It was definitely not made of wood. There is a lot of circuitry that goes behind a computer, and that circuitry, the structure in which it is stored, can be made of wood, but these things have to be conducting, right? Because there are electrical components that are used. So it was not made of wood also. So something out of C and D. What was the correct answer? It was actually D, right? So it uses stored programs for computations. That was an advantage or key feature of this EDVAC computer. Now it was a little different in how it operated from the ENIAC, right? What is the full form of EDVAC? It is Electronic Discrete Variable Automatic Computer. It was one of the earliest electronic computers that was there. It was also built by Moore School of Electrical Engineering at the University of Pennsylvania in United States. Along with OTVAC, it was the successor to ENIAC. Unlike ENIAC, however, it was binary rather than decimal and was designed to be a stored program computer. Now, what does that mean? We'll see. Functionally, EDVAC was a binary serial computer with automatic addition, subtraction, multiplication, program division, and automatic checking with an ultrasonic serial memory having a capacity of 1024 44 bit words, right? 44-bit words. Advanced average addition time was 864 microseconds, and its average multiplication time was 2900 microseconds. So at that time, it was really great, right? These levels were really great. So, which of the following concepts is not a central idea of cybernetics, right? So, what is cybernetics? If we know, we can easily answer this question. So, what are the options that are given to you? Feedback loops, error signals, time travel, or goal-directed behavior, right? So, if you have, gone through this lecture of cybernetics, you will know that it is based on the principle of feedback, right? So, and all of these are concepts that are central ideas of cybernetics except for time travel. So, what is cybernetics? The central idea of cybernetics is the principle of feedback itself, which is the process where effects of a system's action return as inputs, influencing subsequent actions, right? So, if we go back, uh, you can see that feedback loops is a key component of cybernetic error signals. If there are erroneous signals, the subsequent action will also, be changed as opposed to if there was a correct signal that was being transmitted, right? And there is definite goal-directed behavior because the feedback is, changing what needs to be done. So if your input has changed, your output would also change. So the goal because now would have changed. Uh, yeah, this concept is fundamental to understanding how systems regulate themselves through feedback loops, such as a thermostat adjusting temperature or a ship steering in response to environmental changes. Cybernetics explore how these feedback mechanisms operate across various contexts, including a biological, mechanical, and social systems, and is characterized by its interdisciplinary nature, bridging fields like engineering, biology, and social sciences. Right? So, another very common example of a feedback loop that you might have already encountered is, for instance, um, if you are in that lab environment, or there are some such appliances even in our kitchens, wherein you set a temperature and it will attain that temperature. For instance, in a water bath, if you set 70 degree, it will attain 70 degree, and then it will stop heating, right? Once it senses that, okay, now I am 70 degree. And once it stops, obviously that active heating process is not happening anymore. Eventually, it will start cooling. As soon as it starts cooling, it triggers another response that, okay, I'm no longer at 70 degree. Maybe that heating process should start again. And again, that compressor starts. Again, it starts heating, right? The same thing happens with our ACs also now, right? We can set a temperature, and that is also known as that energy saver mode, right? You can set it at a certain temperature. After it has attained that temperature, the compressor will turn off, right? Until it drops like 2-3 degrees again, and or it increases 2-3 degrees again, and then again the compressor will start and begin functioning to bring it back to that temperature, right? So another very simple example of the principle of cybernetic, right? So that is it for the question part for this week. Uh, Let us move to the learning and memory part. So, as I have already told you, last week we had already gone through what are the different kinds of memories and how they are formed. Right, today we are going to talk about what are the molecular mechanisms that take place behind that. Okay, so what I can do is I can see that a lot of you have joined today who were not actually there previously in the previous session. So quickly, I can go through a revision of that, right? Of what we had already studied. And then we can come to this discussion because it is a continuation of what we had studied last week. we are going to study today, right? So we are still going to proceed with our discussion about learning and memory. We are trying to clear our concept further. Uh, yeah. So, hmm. So, since I will upload this lecture to YouTube, and you will probably get the link also. So I'll not go towards all of it. I'll just go through the parts that are important for today's discussion, right? So, for instance, we had discussed what is short-term memory, what is long-term memory, right? Briefly speaking, short-term memory is not what we generally think, right? That, oh, today in the afternoon I met somebody, and for now I remember who that person is, and this is short-term memory. And because I also met somebody who I have known for 10 years, that and I still remember that person, that is long-term memory. So it is not dependent on the time. Like if 15 minutes back I met somebody and I remember who that person is, that is not short-term memory, right? Short-term memory is another term for working memory only, right? So it is more similar to the RAM of the computer. So it means that when you are attentive or when you are alert, whatever your brain is working with, at that moment you remember it, right? But if it has not gone into that storage portion of your brain, or it has not been consolidated into your long-term memory, you will probably forget it, right? For instance, the OTPs that we use, right? So if I get an OTP 2626, right? And I have to put it into another app, I will see that, okay, this is the OTP I got, 2626, and I will probably iterate it twice or thrice in my mind, or I will keep, my focus on it, that I don't forget what this OTP is, because I need to enter it somewhere else. Once I have entered it, 2626, right? I Because OTV is something that you don't, consciously think about remembering, right? So after, say, 5 minutes, because your focus or your attention is lost, and you have not deliberately attempted to learn or remember the OTP, you will forget what the OTP was, right? You might have fabulous memory, but you were not thinking about, or your brain doesn't want to remember that. It was just for that particular span that you required that information, right? So for that particular span, your brain did, retain that information for the purpose of processing, but it never consolidated or converted it into long-term memory, right? Because that is not required by us. So that is basically the difference between short-term and long-term memory. I am not going into the details right now. Short-term memory is selectively transferred to long-term memory, right? Uh, sorry, we'll see how that happens. Today we'll discuss the molecular mechanism behind it as well, right? Uh, I'm just seeing if there is any key point that is left. Yeah. Another important discussion that we had is that once we talk about long term memory can actually be further classified into explicit memory or implicit memory. So we will be talking about both of these today as well. So I'll quickly go through what each of these is, right? Uh, just a moment. Yeah. So what is implicit memory? It is an unconscious form of memory that is evident in the performance of a task and is known as implicit memory, right? Uh, in this case, the memory is automatically recalled. We don't need conscious processing, or consciously we don't need to think about, like how do I do this, right? For instance, different forms give rise to priming, skill learning, habit memory, and conditioning. And it is tightly connected to original conditions under which the learning had occurred. So, implicit memories example could be reading, right? So reading is a skill that I have developed, right? Now, if like I have not retained or like my brain would not have this particular line fed into the brain, right? My brain will have to read every sentence to make out what this is, right? So, but I am not consciously trying to, tap into that area of the brain that understands language and that can actually read it and tell me what the meaning of unconscious is, what the meaning of memory is, or what I taught that day, right? So I am not consciously trying to remember that, right? It is a skill that doesn't, in which some part of the memory is being accessed. That part might have something to do with the words that I know, the vocabulary that I have, etc., right? But it is uh, kind of more autonomous, right? I don't need to particularly go and fetch that memory or think much about it, right? The other kind is explicit memory. It is a deliberate or conscious retrieval of previous experiences as well as conscious recall of factual knowledge about people, places, and things. Right, this type is known as explicit memory. For instance, if I'm learning for an exam, right, writing will come naturally to me. But what I need to write, what I had studied about that subject, I might have to think about, right? So that is this declarative memory or explicit memory, right? Or for instance, I am seeing somebody and I'm trying to recall who this person is. That is explicit memory. Uh, yeah, so we can skip this also for now, and we'll go through this again anyway. So, yeah. Explicit memory actually has, yeah, explicit memory is mediated by at least four related but distinct types of processing, right? One is encoding, the other is storage, then it is consolidation, and then it is retrieval. We'll understand what each of these is, right? So, what is encoding? It is a process by which new information is attended and linked to the existing information in the memory, right? So, the extent of this process is critically important for determining how well the learned material will be remembered, right? So, how well we are perceiving it is how well we might remember it, right? Then comes storage. Storage refers to the neural mechanisms and sites by which memory is retained over time, right? Then comes consolidation. It is a process that makes the temporarily stored, still labile information more stable. It involves expression of genes and protein synthesis that give rise to structural changes at synapses. Again, this is how this happens, is what we are going to study today. Then comes retrieval. Retrieval is the process by which stored information is recalled. It involves bringing back to mind different kinds of information that are stored in different sites. Retrieval of memory is much like perception. It is a constructive process, therefore subject to distortion as much as perception is subject to illusions. So this example I would like to recap, right? Uh, so it is, yeah. So, for instance, there was a scientist who did a series of studies back in 1930s. What he did was he asked certain subjects to read stories and then to retell them. What these subjects actually retold was they recalled stories were shorter, they were more coherent. That is, they were better, had a better flow than the original stories and reflected reconstruction and condensation of the original material. So these subjects were unaware that they were editing these original stories and often felt more certain about the edited part than about the unedited part of the retold story. And they were actually not confabulating. So what it means is that for instance, somebody told me a story, right? And once I'm trying to recall it or retell it, my brain will try to make sense of that story, right? So for instance, somebody came and told me two sentences that uh, Shivam got a new bat and Shivam got a new bat Shivam is also a cricketer, and uh, he is not going to play the next match, right? But he's not going to play the next match. Something, some three statements, right? Shivam got a new bat. Shivam is a cricketer. Um, and he's not going to play the next match, right? So, for instance, these were the three statements that were told to me or that were told to you, right? After a certain point of time, if you have to retell this to somebody, your brain will naturally try to make sense of what was told to you, right? So, you might say that there's a cricketer known as Shivam. Uh, he just bought a bat, but he'll not be playing tomorrow. Probably because he got injured, right? Something your brain might add, right? So just to make sense of why is he not playing tomorrow, right? You right now I'm deliberately doing it, so it is not that precise, right? But you might not be aware that you added a fact or you, modified the statement a little bit, but that was not you deliberately doing it. It was your brain trying to make sense of the story that you are retelling, right? So, to make it more coherent. Yeah. Okay. So, the rest of it probably you can watch in the lecture that I will upload, right? Oh, I got confused. Exactly 63 slides last time, exactly 63 this time. Yeah. So on a very superficial level, for now we have revised what is short-term memory, what is long-term memory, what are certain subtypes of long-term memory, how does storage occur, uh, or what are the key features of this long-term memory, right? The brain regions that are involved in this, that part I have skipped right now. There were certain subtypes of these memories as well. That also for now I have skipped. You can go back and you can watch that lecture, right? So let us begin today's discussion. When we talk about implicit memory, so that is why we went back and we actually understood what implicit memory was, right? Because this is a continuation of that subject. So, implicit memory supports perceptual priming, right? So, what this means is implicit memory stores forms of knowledge that are typically acquired without conscious effort and which guide behavior unconsciously, right? So, we just studied that what was implicit memory? It was a kind of memory which comes with, when we attain a skill, when we learn something new, for instance, if I learn how to drive or how to play a guitar, right? So that too, and I try to drive again or play a guitar again. So that will be implicit memory coming into picture because, by default, I'll be able to do that. By default, I can talk, right? Every time my brain doesn't have to go and fetch that, oh, my mother taught me that we have to say hello once we meet somebody, and my teacher in school, she taught me this particular sentence, and that teacher taught me that particular sentence. So that is not how my brain is working, right? That is the what implicit memory does, right? So priming, what is priming? That is new, right? Priming is a type of implicit memory that operates in amnesic patients as well as healthy patients, suggesting that it does not depend on the medial temporal lobe structures at all, right? Because amnesic patients generally have resected parts or completely resected medial temporal lobes. So even in those patients, this priming works well. So it has nothing to do with this particular region, is what we are sure about. There are two types of priming that have been proposed. One is conceptual priming and the other is perceptual priming. So what is conceptual priming? It provides easier access to task relevant semantic knowledge because that knowledge has been used before. That is, it is correlated with decreased activity in left prefrontal regions that subserve initial retrieval of semantic knowledge. In contrast, what is perceptual priming? It occurs within a specific sensory modality, and according to Tulving and Schatzer, it depends on cortical modules that operate on sensory information about the form and structure of words and objects. So we'll come to examples and then you'll understand better, right? So we are saying that it is supporting perceptual priming instead of conceptual priming, right? So what does this mean? Perceptual learning involves a shift from cognitive to autonomous stages that use different neural pathways. So, as I said, uh, there is some cognitive pathways in which cognitive stages in which your brain is actively trying to think in a certain way. There are multiple things that you are consciously doing in order to understand what is happening, right? Or what we are trying to do or what we are trying to remember, right? Autonomous is more automatic, right? So if, for instance, subjects are asked to read mirror reverse text, something most people rarely encounter, right? We generally don't see text written like this, right? So if I have to read this particular these particular mirror inverted text, uh, my brain might take a little while if I'm seeing it for the first time, right? But by now I have probably seen it thrice or four times maybe, right? So I can immediately read that this first word is chair and the other is apple, right? But if you are seeing it for the first time, it will take you a little longer than it took me to read what that word is, right? Or even if a new word which is inverted will be given to me, it will still take me lesser time than somebody who is seeing that kind of mirror inverted text itself for the first time, because now my brain is trained for it, right? So, for instance, you can see before training, this area was active in patients, right? But after training, this area was not that active, while another area became more active, right? So, there is some kind of a change in how, this perception is occurring. So prior to training individuals, they rely on the parietal cortex, which has this red arrows, and to a lesser extent to the inferior temporal cortex over here. After extensive training, the processing pathways involved in task appear to be different, however. Right? Individuals rely less on the parietal cortex and more on the inferior temporal cortex over here. Right? So these results reflect a transition from having to mentally rotate the mirror reversed words to the ability to read directly the reversed letters. Right? So that is what is happening. Different neural processes are involved once skilled performance moves out from the cognitive to the autonomous stage. So similar neural changes have been observed in imaging studies of motor and visual motor skill learning. So this was one example. There are other examples which also prove that implicit memory actually supports perceptual kind of priming instead of consciously thinking, right? So implicit memory can be associative or non-associative. So what does this mean? Uh, so, post-human studies, there were studies that were done on animals as well, right? That is when this concept of associated and non-associative memory actually arose. So, non-associative learning is when an animal learns about the properties of a single stimulus at a time. Associative learning is when animal learns about the relationship between two different stimuli or between a stimulus as well as a behavior, right? So, non-associative learning, when we talk about it, results when a subject is exposed to once or repeatedly to a single type of stimulus, right? Two forms of non-associative learning are common in everyday life. One is habituation, the other is sensitization. They are actually the opposites of one another. What is habituation? It is a decrease in response occurring when a benign stimulus is presented repeatedly. So, for instance, um, if I hear a loud bursting, like a blast like sound, right? I will be very alarmed. I will probably, I'll be a little shocked. I'll have some kind of a reaction to it, right? I might go out and check what just happened, right? Was it a cracker, or was it some, electrical, circuit explosion that happened, or was it something else altogether, right? So I might be a little taken aback. I might be shocked. I might go and check what it was. That is a normal response to a normal stimuli, right? To an abnormal stimuli, actually. But if the same thing happened on, say, Diwali or the Shera, right? I heard a blast sound, but I keep hearing a blast sound because people are, bursting firecrackers outside. So I'll be habituated to it. I will not have the same kind of a response, right? So that is when there is a decrease in response because of a continuous stimulus that is being presented, right? That is what is habituation. Sensitization is actually opposite to that. Sensitization is also known as pseudo conditioning. It is an enhanced response to a wide variety of stimuli after the presentation of an intense or noxious stimulus. Moreover, sensitizing stimulus can override the effects of habituation, which is known as dishabituation. So, first, let us understand by another example what is sensitization, right? For instance, um, how I'm thinking of an example that will be easy to explain. Yeah. For instance, the taste of cinnamon is very mild, right? Specifically, if you use it with other spices, right? Say you are a chef, and you are learning how to recognize tastes or different flavors from what you are eating, right? So you have to be sensitized to each of the tastes, right? Normally, you might just, take a piece of paneer from the gravy and just eat it like that, not bothering what is there in that paneer, what is not there. It is just tasting nice. That is all for you, right? Uh, but say you are a chef, you are trying to recreate a certain dish, right? So you might have undergone training for precisely that kind of a thing that needs to be performed, right? So for that, you might have sensitized your taste buds to very minor, changes, or very minor, levels of certain spices, certain chemicals, certain tastes in the food itself, or certain odorants also associated with food, right? So, for instance, cinnamon again, in that group, making out something like cinnamon might be difficult for a novice person, somebody not associated with cooking that much. But for a chef, a cook, it might be really simple, right? Because they have sensitized themselves to that stimuli multiple number of times, and now they can make it out even in very small amounts. So this is a good example of a sensitization that I actually gave. Generally, sensitization happens because of an intense or noxious stimuli, right? So what does that mean? For instance, once I had an encounter with a poisonous mushroom, right? So again, it is not very likely that I will even, go near that kind of a mushroom, or maybe its smell itself will cause an aversion, because now I'm sensitized that, okay, this is something that is going to cause a problem, right? So I'm sensitized to that situation. That is what is sensitization. Then comes conditioning, right? So classical, there are two kinds of conditioning. One is operant conditioning, one is classical conditioning. First, we are going to understand classical conditioning. So before this, what we were looking at, non-associative learning, right? Non-associative learning included habituation as well as sensitization. Associated learning includes classical conditioning and operant conditioning, right? So first, let us understand classical conditioning. Now, this is something that uh, we have all come across, even if not in the terms of science, I feel. You might have all heard it in some that this was a experiment that was done, maybe in some animal behavior, maybe in some psychology lecture, because there are multiple fields in which this example is actually used. So, it was first described at the turn of century by Russian physiologist Ivan Pavlov, right? So, this was actually also known as a Pavlovian experiment or Pavlovian conditioning, it is known as, right? Essence of classical conditioning is the pairing of two stimuli, right? That is the essence of it. So this was that famous Pavlovian experiment. What it says is that when a particular subject, in this case a dog, is not conditioned, right? Before conditioning, the food that is given to the dog is an unconditioned stimulus, right? So when the food is shown to the dog starts salivating, right? And when there is another stimulus which has nothing to do with the food at this point, it is a neutral stimulus. In this case, it was a bell. Before conditioning, when it was rung in front of the dog, there was no effect that was seen, right? During conditioning, what happened was that for a few consecutive days or for a few consecutive meals, what Pavlov actually did was he showed the dog the meal of the dog. Along with that, simultaneously he would always ring the bell, right? So the dog would salivate, right? After conditioning, what happened? Even when the meal of the dog was not present and only the bell was rung, right? The dog actually started salivating, right? Why did this happen? Because during conditioning, this particular neutral stimulus, it became associated with him getting the meal. So whenever he would get a meal, he would also hear the bell ring, right? So by default, he associated the two things together, right? So if he would hear only the bell ring, he would assume that he's going to get the meal, right? Because that is how he was conditioned during some time. So this is the classical conditioning, and this was the classic experiment that was done by Pavlov, right? One of the classic experiments. So one way of explaining the conditioning is that the repeated pairing of the conditioned stimulus and unconditioned stimulus causes the conditioned stimulus to become an anticipatory signal for the unconditioned stimulus. With sufficient experience, an animal will correspond to conditioned stimulus as if it were anticipating the unconditioned stimulus, right? Probability of occurrence of a conditioned response decreases if the conditioned stimulus is repeatedly present without the unconditioned stimulus. This process is known as extinction, and it is correct also, right? So, if continually the bell would, like every time again, Pavlov would come and ring the bell and not give the dog the meal, eventually the dog will stop responding to or stop salivating to just the ringing bell, right? So there would be an uncoupling or unconditioning that would happen, and this process would be known as extinction. And this is something that is evolutionarily also advantageous to us, right? So we because we will not spend the same type of energy in that particular response or in anticipating a particular thing if that is not happening. So it is evolutionarily advantageous, the process of extinction in this term, right? Classical conditioning and perhaps all form of associative learning enables animals to distinguish events that reliably occur together from those that are only randomly associated, right? So then operant conditioning, which is the other kind of associative learning, involves associating a specific behavior with a reinforcing event, right? So I'll come to what operant conditioning is. Please give me a moment. Yes. Okay. So operant conditioning involves associating a specific behavior with a reinforcing event, right? So this is another experiment that you might have come across if you have ever studied the subject animal behavior, right? So in that is where I had first come across operant conditioning per se, right? Classical conditioning that we have been, given the example of for a while now in different subjects in different scenarios, right? So, a second major paradigm of associative learning, discovered by Edgar Thorndike and systematically studied later by B.F. Skinner and others, is operant conditioning, which is also known as trial and error learning. So, in this, what they used to do was there was something known as Skinner box, right? So, it was named after this B.F. Skinner. So in the Skinner box, what used to happen is that they used to, test with different kinds of animals, with mice, rat, even dogs, cats, etc. Right? So what they used to do is, in this box, there would be a negative reinforcer and a positive reinforcer that used to be present. Right? Positive reinforcer in this case is the food pellet and the water. Okay, in this particular one, water is not there. In this particular one, positive reinforcer is the food pellets, right? So if the mice presses this lever, it will get food pellets, right? Depending upon which light is blinked. So for instance, a blue light, whenever blue light blinks, the mouse should press this lever and it will get food, right? And whenever yellow light rings, uh, 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 blinks, the mouse should not press the lever. If when the yellow light blinks, the mouse presses the lever, it will get a shock because to at the bottom of this particular box, it was connected to a shock grid, right? So now the when the mouse is going inside, it doesn't know that this is what is going to happen, right? So with trial and error, the mouse learns this, right? So it would happen that yellow light would blink and he would press the lever, and if yellow light blinked and he pressed the lever, he would get a shock, right? Then again, maybe yellow light blinked again, he pressed the lever, so he might get the shock, right? So he might get averse to pressing the lever. But at the same time, maybe after one or two trials, they blinked this blue colored light. And at that time, maybe accidentally or maybe knowingly, the mouse again pressed the lever. And he got this food pellet, right? So he got something to eat finally after getting all that shock, right? And again, this happened again, he got a food pellet, right? And again, it was switched and the yellow light was blinked. Now the mice got used to pressing the lever and getting the food pellet. Again, he pressed the lever, but he got shock. So eventually, the mouse understood that okay, if this light is blinking, I should not press the lever, otherwise, I would get shock. If this light is blinking, I should press the lever, I will get food. Right? So it is a trial and error based behavior or conditioning, right? So that is why there is a negative reinforcer, which is a shock, and a positive reinforcer, which is the food pellets. I hope it is clear, right? So if there are any doubts, please stop me in the middle. I'll try to clear that, right? So, if we think of classical conditioning as the formation of a predictive relationship between two stimuli, the conditioned stimulus and unconditioned stimulus, the operant conditioning can be considered as the formation of a predictive relationship between an action and an outcome. If I press the lever, I might get food, but if I press the lever, I might also get shock, right? So, that kind of a predictive relationship it is telling. Unlike classical conditioning, which tests the responsiveness of a reflex to a stimulus, operant conditioning tests behaviors that occur either spontaneously or without an identifiable stimulus. So there are two forms of long-term memory which involve different brain systems, right? So, implicit memory that we studied about earlier, which is a non-declarative memory, we don't have to consciously recall it, involves these particular regions such as neocortex, striatum, amygdala, cerebellum, reflex pathways, depending upon which kind of implicit memory we are talking about, right? Explicit memories, which are, facts and events that we are trying to remember, the semantic and episodic memories, they are recalled through the medial temporal lobe. This is what was affected in the patient HM. It was resected in the patient HM. That is why he did not have any explicit memory whatsoever, right? At least the newer ones. Okay. So these are this is a medial temporal lobe. This is striatum, neocortex, amygdala, cerebellum, and reflex pathways over here, right? So the storage of implicit memory involves changes in the effectiveness of the synaptic transmission, right? So how the synapses are, interacting with each other is what is going to affect how implicit memory is stored. Habituation results from activity-dependent presynaptic depression of synaptic transmission. So we went through two kinds of these non-associative memories. One was habituation, the other was sensitization. First, let us understand about habituation. So it results how habituation occurs. It results from an activity-dependent presynaptic depression of synaptic transmission. What it means is that it occurs when an animal learns to ignore a novel stimulus, right? So, for instance, I gave you an example of that blast-like noise that I heard, right? If it happened right now, I would probably be alarmed by it. I might try to check what happened, right? But at the same time, if it happened around, say, Dashera, Diwali, some event, some time of the year when firecrackers are a big thing over here, everybody's bursting firecrackers around you, it will not alarm me at all, right? I will learn to ignore that stimulus. So the physiological basis of habituation was first investigated by Charles Sherrington while studying posture and locomotion in the cat. Sherrington observed a decrease in the intensity of certain reflexes in response to repeated electrical stimulation of the motor pathways. He also suggested that this decrease, which he called habituation, is caused by diminished synaptic effectiveness in the stimulated pathways. Right? Habituation was later investigated at cellular level by Alden Spencer and Richard Thompson. What they did was they found that close cellular and behavioral parallels between habituation of a spinal flexion reflex in cats and habituation of more complex human behaviors as well. They showed that during habituation, strength of input from local excitatory interneurons onto motor neurons in spinal cord is actually decreased. So they wanted to understand this, but at the level of higher organisms, there were so many other levels. There were inhibitory neurons, there were interneurons, there were multiple dendritic branches. So it was a very complex system. So they wanted to study it in a less complex system, which is where Aplysia, which is a marine mollusk, came into picture, right? So Aplysia californica has simple nervous system of about 20,000 CNS neurons, which proved to be an excellent system for studying implicit forms of memory. Aplysia has a repertory of defensive reflexes for withdrawing its respiratory gill and siphon, a small fleshy spout above the gill, which is used to expel seawater and waste. So even if you mildly touch the siphon, it elicits reflex withdrawal of both the siphon and the gill. With repeated stimulation, these reflexes eventually stop occurring and they habituate to the situation. So this is aplysia, right? This is the respiratory organ or the gill. This is the siphon that we are talking about. This is the shells, and this is the tactile stimulus. So if for instance, this stimulus is applied on the siphon, the gill will retract, right? So, for instance, like this. So, this is normally how the gill is, this dotted line. And if there is a stimulus, the gill will retract into this much, right? This is if it happens initially, but if there is a continued stimulus that is applied to this siphon, it will get habituated to this particular place and the gill withdrawal will reduce or it will not occur at all. So this simplified circuit shows key elements of the gill withdrawal reflex as well as sites involved in habituation. Approximately 24 mechanoreceptor neurons in the abdominal ganglion innervate the siphon skin over here. These skin sensory cells make excitatory synapses onto the cluster of six motor neurons that innervate the gill as well as on interneurons that modulate the firing of motor neurons. So over here and here, it will give an excitatory synapse, right? Touching the siphon leads to withdrawal of gill like this, right? But repeated stimulation of the siphon sensory neuron leads to a progressive depression of synaptic transmission between sensory and motor neurons, seen as a decrease in size of motor neuron EPSP with no change in the action potential in the presynaptic sensory neuron. So you can see the end plate potential, it is also decreasing and the action potential is also not there anymore, right? You can see over here at the level of synapse also the neurotransmitter level is also reducing. So in a separate experiment, repeated stimulation of siphon results in a decrease in gill withdrawal. 1 hour after repetitive stimulation, both the EPSP and gill withdrawal have recovered. Habituation is now known to involve a decrease in transmitter release at many synaptic sites throughout the reflex circuit, right? As you can see over here. So, what reduces the effectiveness of synaptic transmission between the sensory neurons and the postsynaptic cells during repeated stimulation? When quantal analysis was done, it was revealed that the amount of glutamate, which is a neurotransmitter, from the presynaptic terminals of sensory neurons actually decreases. That is, fewer synaptic vesicles are released with each action potential in the sensory neuron, and sensitivity of postsynaptic glutamate receptors does not change at all. Because of the reduction in transmission, occurs in activated pathway itself and does not require another modulatory cell. The reduction is referred to as homosynaptic depression. This depression lasts for many minutes. An enduring change in the functional strength of synaptic connections thus constitutes the cellular mechanism mediating short-term habituation. Then comes long-term habituation. So what is it? Anatomical studies indicate that long-term habituation is caused by decrease in the number of synaptic contacts between sensory as well as motor neurons, right? So what happens over here? There is sensitization which involves presynaptic facilitation of synaptic transmission. When the animal repeatedly encounters a harmless stimulus, its responsiveness to the stimulus habituates. In contrast with a harmful stimulus, the animal typically learns fear. It responds vigorously not only to harmful stimulus but also to the other concurrent stimulus, even the harmless ones. As a result, defensive reflexes for withdrawal and escape become heightened. This enhancement of reflex responses is called sensitization. So, sensitization actually involves presynaptic facilitation of synaptic transmission. Uh, you can see over here, there's a depression of synaptic potential by long-term habituation, as you can see, right? So, initially, this is what happened, and after habituation, this is the kind of response you see, right? Comparison of action potential and synaptic potentials in the sensory and motor neurons, respectively, and an untrained animal and the one that has been subjected to long-term habituation. In habituated animal, 1 week after training, no synaptic potential occurs in the motor neuron in the response to over here. See the action potential or synaptic potential is no longer occurring in response to sensory neuron action potential, right? So sensory neuron, there is still an action potential, but motor neuron, there is no response that is occurring, right? That is what is habituation. Also, in this case, you can see the inactivation of synaptic conditions by long-term habituation. So eventually it will go down, right? So that is what it is being habituated to. The mean percentage of sensory neurons making physiologically detectable connections with motor neurons in habituated animals is decreased at 3 points in time after long-term habituation training. Yeah. Sensitization and dishabituation result from enhancement in synaptic transmission at several connections in the neural circuit of the gill withdrawal reflex, including connections made by sensory neurons with motor neurons and interneurons, and the same synapses depressed by habituation. In aplasia, when we are considering the same synapses that are weakened by habituation through homosynaptic processes can be strengthened again by sensitization also through heterosynaptic process that depends on modulatory interneurons activated by the harmful stimulus to the tail, right? So this is what happens. Sensitization involves presynaptic facilitation of synaptic transmission. So, over here, you can see this is gill sensitization, similar to what we had talked about after sensitization. In habituation, what was happening? That there was no motor response in the same action potential of the sensory response also. Right over here, you can see there is an increased motor response in the same action potential from the sensory neuron. Sensitization of gill withdrawal reflex is produced by applying a noxious stimuli to another part of the body, such as tail. A shock to the tail activates tail sensory neurons that excite facilitating or modulatory interneurons, which form synapses on the cell body and terminals of the mechanoreceptor sensory neurons that innervate the siphon, right? Through these axonic synapses, the modulatory interneurons enhance transmitter release from siphon sensory neurons onto their postsynaptic gill motor neurons, thus enhancing the gill withdrawal. And presynaptic facilitation results in part from a prolongation of the sensory neuron action potential, right? The basic was what we talked about when we saw the diagram, right? Sensitization involves presynaptic facilitation of synaptic transmission. So, at least three groups of modulatory interneurons are involved in sensitization as we know it. The best studied group out of this is a group which uses serotonin as a neurotransmitter, right? So, the serotonergic interneurons form synapses on many regions of the sensory neurons, including the axoaxonic synapses on the presynaptic terminals of the sensory cells. The serotonin released from the interneurons after single tail shock binds to a type of receptor in the sensory neuron that is coupled to a stimulatory G protein that increases activity of adenylyl cyclase, right? What it does is it will activate the secondary messenger, if you remember signaling. So action produces a second messenger cyclic adenosine monophosphate CAMP, which in turn activates the CAMP dependent protein kinase or PKA. Serotonin also activates the second type of GPCR that leads to hydrolysis of phospholipids and activation of protein kinase C. So PKA, PKC both are activated, right? What they do is protein phosphorylation is mediated by both these protein kinases, and it enhances release of neurotransmitter from the sensory neurons. So it could occur via any of these pathways actually, right? So there are five different pathways that are shown over here. This is one through this cyclic AMP that and PKA pathway. This is the second one through that another GPCR PKC pathway. Okay, this is just the two. The five pathways come much later. We'll come to that. So these were the two pathways that we talked about. The first one is a facilitating interneuron releases serotonin or 5-HT, which is the technical name for serotonin, which binds to metabotropic receptors in the sensory neuron terminal. This action engages the G proteins, which in turn increases activity of adenylyl cyclase over here. Adenylyl cyclase converts ATP to EMP to cyclic EMP, which binds to the regulatory subunit of pKa, thus activating its catalytic subunit. The catalytic subunit phosphorylates certain potassium channels over here, thereby closing the channels and decreasing the outward potassium current. This prolongs the action potential, thus increases influx of calcium through voltage-gated calcium channels and thereby augmenting transmitter release. Right, so calcium influx is there, due to which these vesicles they will go and they will merge with the post presynaptic terminal and release a neurotransmitter outside. So this was point one or pathway one. The second pathway is where serotonin binds to a class of metabotropic receptor that activate the GQ11 a class of G protein that enhances the activity of phospholipase C, PLC, right over here. The PLC activity leads to production of diacylglycerol, which activates protein kinase C over here. Phosphorylation of presynaptic proteins by PKC results in mobilization of these vesicles containing glutamate from a reserve pool to a releasable pool at the active zone, increasing the efficiency of the transmitter release. Then comes classical conditioning. So, classical conditioning of fear basically involves coordinated pre and post synaptic facilitation of synaptic transmission. Right? Classical conditioning is a more complex form of learning than what we were already seeing. Right? An initial weak conditioned stimulus becomes highly effective in producing a response when paired with strong unconditioned stimulus, such as presentation of food and ringing of a bell, right? For classical conditioning of aplasia, gill, withdrawal reflex, a weak touch to siphon serve as the conditioned stimulus, while a strong shock to the tail serves as unconditioned stimulus, right? So this is the unpaired pathway, right? In the unpaired pathway, there is a stimulation of the siphon and there is a shock given to the tail, but they are not coupled together, right? So before training and after training, you can see the gill motor neuron EPSP is decreasing over here. Siphon is stimulated by a light touch and tail is shocked, but the two stimuli are not paired in time. The tail shock excites facilitatory interneurons that form synapses on the presynaptic terminal of the sensory neurons, innervating the mantle sheath and siphon. This is the mechanism of sensitization. The pattern of unpaired stimulation during training, and under these conditions, size of the motor neuron EPSP is only weakly facilitated by the tail shock. In this example, the EPSP actually decreases slightly despite the tail shock because repeated unpaired stimulation of the siphon leads to a synaptic depression, right? There is a paired pathway as well, wherein the shock that is given to the tail and the siphon being stimulated, they are actually coupled. So, tail shock is paired with the stimulation of siphon. Siphon is touched, that is conditioned stimulus or CS, and immediately prior to shocking the tail, which is the unconditioned stimulus or US. As a result, the siphon sensory neurons are primed to be more responsive to input from the facilitatory neuron in the unconditioned pathway. This is a mechanism of classical conditioning in both amplifies the response of the conditioned pathway and restricts the amplification of that pathway, right? So you can see over here the response is amplified instead of going down below the EPSP line. Recordings of EPSPs in an identified motor neuron produced by the siphon sensory neurons before training and 1 hour after training. After training, EPSP in motor neuron produced by paired sensory input is considerably greater than either the EPSP before training or EPSP following unpaired tail shock. This produces a more vigorous gill withdrawal as well, right? Then long-term storage of implicit memory involves changes in the chromatin structure and gene expression, which is mediated by CAM, PKA, CREB pathway, right? So this is the molecular mechanism. Uh, we can go through it. Probably it will be the last point we'll go through. So whatever is left, we'll take up in the following lecture, right? So for now, in all forms of learning, practice makes perfect, right? Repeated experience converts short-term memory into long-term form. So what happens during this consolidation? Long-term sensitization, like short-term form, requires protein phosphorylation that is dependent on increased levels of cyclic AMP. The conversion of short-term memory into long-term memory is called consolidation, which requires synthesis of messenger RNAs and proteins in the neurons in the circuit. That specific gene expression is required for long-term memory. The transition from short-term to long-term memory depend on the prolonged rise in CAM that follows repeated actions of applications of serotonin. So this leads to prolonged activation of protein kinase A, allowing the catalytic subunit of kinase to translocate into nucleus of the sensory neurons. It also leads indirectly to activation of second protein kinase, mitogen activated protein kinase or MAPK, a kinase commonly associated with cellular growth. Within the nucleus, catalytic subunit of pKa phosphorylates and thereby activates the transcription factor CREP1 or CAMP response element binding protein 1, which binds a promoter element called CRE or CAMP recognition element. So these are this is that five pathway that I was talking about. Long-term sensitization of gill withdrawal reflex of aplasia following repeated tail shocks involves long-lasting facilitation of neurotransmitter release at the synapses between sensory and motor neurons. Right? What happens over here? Long-term sensitization of gill withdrawal reflex leads to persistent activity of pKa over here, uh, resulting in growth of new synaptic connections. Repeated tail shock leads to more pronounced elevation of CAMP or cyclic AMP, producing long-term facilitation and that outlasts the increase in the CAMP and recruits the synthesis of new proteins. Inductive mechanism is initiated by a translocation of protein kinase A to the nucleus, which is pathway 1 over here. Uh, Protein kinase 1 to the nucleus over here, right? Yeah. Where pKa phosphorylates the transcriptional activator CREB. So CREB is the transcriptional activator. PKA will go and it will phosphorylate it. Uh, then CREB1 actually binds CAMP regulatory elements over here, which are CRE, which are located in upstream region of several CAMP-inducible genes, activating gene transcription, right? So once this gene transcription has activated, PK also activates the mitogen activated protein kinase MAPK, which phosphorylates the transcriptional repressor CREP2 over here, thus removing its repressive action. One gene activated by CREP1 encodes the ubiquitin hydrolase, a component of a specific ubiquitin proteasome that leads to proteolytic cleavage of the regulatory subunit of pKa, resulting in persistent activity of pKa even after CAMP has returned to its resting level over here. CREF1 over here also facilitates expression of transcription factor CEBP uh, over here, right, which leads to expression of a set of unidentified proteins important for growth of new synaptic connections. So that was the fifth pathway. Then how are there modifications at the level of DNA folding that occur, right? So there is a regulation of histone acetylation by serotonin, CREB1 and CBP, right? So generally, when the histones are DNA is wrapped around histones, right? So when the DNA is in this wrapped position, it is transcriptionally blocked. It is not actively transcribing, but for it to transcribe, there has to be loosening of its folds over here so that transcription can be permissible. Under basal conditions, the activator CREB1, here in complex with CREB2 over here, occupies the binding side of CRE, right, which is a camp recognition uh, element within the promoter region of its target genes. In the example shown here, CREB1 binds to CRE within the CEBP promoter over here. In the basal state, CREB1 binding is not able to activate transcription because starter box, the core promoter region responsible for recruiting RNA polymerase 2 or Pol2 during transcriptional initiation is inaccessible because DNA is tightly bound to histone proteins in the nucleosome, right? But later what happens? Serotonin activates pKa, which phosphorylates CREB1, indirectly enhances CREB2 phosphorylation by MAPK, causing the repressor to dissociate from the promoter. This allows CREB1 to form complex at the promoter with CREB binding protein, CBP, right? Activated CBP, this is recruitment of CBP. Then activated CBP acetylates specific lysine residues of the histones, causing them to bind less tightly to DNA. Along with other changes in the chromatin structure, acetylation facilitates the repositioning of the nucleosome that previously blocked access of Pol2 complex to the Tata box. This repositioning allows Pol2 to be recruited to initiate transcription of the CEBP gene, right? So this long-term habituation leads to loss of synapses and long-term sensitization leads to increase in number of synapses. When measured either on day one or 1 week after training, number of presynaptic terminals or boutons, these are the terminal ends which are known as boutons, um, relative to control levels is increased in sensitized animals and reduced in habituated animals, right? So, this is a normal condition. After long-term habituation, postsynaptic, the terminals, they reduce, and after long-term sensitization, they increase, right? The number of synapses will increase. The drawings below the graph illustrate the changes in number of synaptic contacts, swellings, or varicosities in sensory neuron processes are presynaptic terminals itself, right? This is a microscopic image of the same, wherein fluorescence images of axons of a sensory neuron contacting a motor neuron in the culture. And 1 day after 5 brief exposures to serotonin, you can see result in increase in varicosity stimulates the synaptic changes associated with long-term sensitization. Prior to serotonin application, no presynaptic varicosities are visible in the outlined area. After serotonin, the growth of several new varicosities is apparent, indicative of formation of new synapses. Boutons can be seen as the arrows in the right. These are the nerve terminal, swollen nerve terminals or boutons. Um, and for now, that could be the end of, not the end of, this is where we again take a break in learning and memory, our discussion about learning and memory, right? So it is a very big topic actually. So yeah, I think one more or half more session we'll spend in learning and memory, because anyway the latter part of your course has more to do with the Nobel laureates and the works that they have done, and that is being talked anyway in pretty much detail. So over here we'll try to cover other kinds of things that we have probably not covered up in our course, right? So we'll talk a little bit more in detail about this learning and memory. Then, as I told you, we'll go on to different kinds of methods of analysis of the brain. So that is what we are going to study, right? So, yeah, thank you for joining. I hope you understood. I'll upload the recording of the previous session as well as this one, and you'll probably get the links from NPTEL. And if there's a problem, please let me know. If there's a doubt, please let me know. I'll try to solve it. Yeah. Okay. So I'll end the session for now. Thank you all for joining.